Okay. All right, let's go to our next part. So again, we are talking about structures and functions of macromolecules. First, we talk about proteins when we talk about nucleic acids. And so this is about structures and functions of nucleic acids. And in the part one, we talk about the component of nucleic acids, which is different nucleotide. And then now in this part, I want to talk about the actual nucleic acids, which is DNAs and RNAs. So proteins are like the actual functional carrier. Proteins can do a lot of things. Protein can defend against pathogens, can be hormones, can be nutrients carriers. Protein can do a lot of things, but DNAs and RNAs are so-called information molecules. They do not do the amazing task. What they are information. They carry informations. DNA carry inheritance information. Inheritance means like how you look like your parents. Okay, how any baby look like their parents, because they get some information from their parents. And this information can direct the cell to grow, to differentiate, and eventually make a person that look like somewhere a mixture of the parents. And that's inheritance information. And that information is carried from your parents to you by this molecule called DNA. Okay, so DNA is, so if we use analogy from computer, DNA encodes information in itself. DNA encodes that information. And RNA is, by analogy to computer, RNA decodes this information. So first you need to have the information, then you need to express this information in the form of proteins. And that is done by RNA. So DNA is a molecule for encoding, while RNA is a molecule for decoding that information. And protein is a molecule that does the actual work. Okay. So so in this part, first we talk about DNA, then we talk about RNA. First, DNA is the molecule that carries inheritance information, and that is the only function we know about DNAs. Protein can do a lot of things, RNA can do a lot of things, but DNA only does one thing. That is, they carry inheritance information. And people know, well, there was a lot of debates in around 1930s, like whether, well, people know there's the inheritance because everyone look like their parents, but is this done by protein or does this done by DNA? There was a lot of debate in 1930s. And someone called Griffiths did this experiment that confirmed that it is DNA, not anything else, that carries the inheritance information. This is what he did. There are two types of bacteria. One is normal wild-type bacteria. They are smooth. They have the smooth surface. They are virulent, means they are toxic. When you inject this bacteria to mice, the mice die. Okay, and he accidentally isolated a mutant type of the virus. This virus, the surface is very rough, and when you inject this bacteria to mice, mice can live happily. So this virus, it's a mutant. It cannot kill mice anymore. Okay. And if we heat, if we increase temperature, if we boil this virulent wild-type virus, okay, we can kill all the bacteria, and the mouse can still live. Okay, this is everything we know. And the interesting thing is this: when we mix the heat-killed virus, the dead virus, with the mutant virus, So the dead virus cannot kill animals. The mutant virus cannot kill animals. But when we mix them together, and somehow the mutant virus acquired something from the dead bacteria, now the mouse just die. Okay. After this mixture, receiving this mixture, the mouse just die. Okay. So then he did a final experiment. So what exactly in the dead bacteria can cause the mutant virus, can cause the mutant bacteria now become lethal to this animal? So he basically, to do that, basically you can isolate DNAs, you can isolate proteins from the dead bacteria and mix that with the mutant bacteria and see which one can now kill the mouse. Okay, and what he found is it's the DNA part, not the protein, that actually can kill the mouse. And this one is done by Dr. Avery. So this actually, he did not get Nobel Prize. So they're saying that but he does deserve one. So people ask great scientists, do you think like who deserves to get Nobel Prize but didn't get it? That's him, because he is the first one that proved that it is DNA carries the information, inheritance information, not the not protein, not something else. And in 1950s, there's another experiment called Hershey-Chase experiment. So what they did is this. They used a bacterial virus. These are viruses that infect bacteria. And this virus has this kind of structure. It has a coat. So the outside of this virus is protein, and inside is nucleotides. So the virus looks like this. It's coated by proteins. So outside is protein. And the inside is probably DNA. And they did a radioactive labeling. So the outside, the protein, the DNA, they labeled with S35. And they labeled the protein with P32. Do you know how is this possible? Because the elements in protein is COCHONS. The elements in DNA is CHONP. Okay. Do you know why proteins has sulfur? There are two amino acids that has sulfur: cysteine and methionine. Okay. And DNA has a lot of phosphate because it's connected by the 3' to 5' phosphodiester bond. So DNA has a lot of phosphate group. Okay. So you can use P32 to label DNA, S35 to label proteins. And then after that, he basically attached the bacteriophage, the bacterial virus, to bacteria and see which get inside. Okay. And see which get inside, which left out. So when he used P32, when he used the P32 to label the inside, he found that the P32, the radioactive thing, get into bacteria, and the non-radioactive protein coat is left. And when he labeled the protein coat, then he found that the bacteria itself is not radioactive, and its coat itself gets left out. So the conclusion is when a bacteria gets infected by its virus, the virus only injects DNA into bacterial and then transform the bacterial into something else and replicate more bacterial. Replicate more virus. So only DNA is needed to get into bacteria to make more virus. The protein code, the outside, the protein code is not required. So this is another experimental proof that DNA carries the information. The protein is not required for the information. Okay. And then after that, there's a lot of efforts going on, like what the DNA actually look like. Okay. DNA, if it's so important, if it carries the information, what does it actually look like? And this is done by 4 different scientists in Cambridge, in England. So the 2 most important are James Walton and Francis Crick. You probably heard of them. And then another 2 scientists, one is called Rose Franklin. Franklin is a female scientist. And at that time, female scientists are not very respected in England. And Franklin and another scientist called Wilkins. So Wilkins thinks Franklin worked for him, but Franklin didn't think so. Franklin thought she works independently. Okay. And so one day, so Franklin is a crystallographer. Franklin make crystals of DNA and try to use X-ray diffraction to calculate what the structure of DNA is. And one day, Franklin got a photo of X-ray diffraction look like this. It's called photo 51. And so Wilkins gave this photo to James Walton and Francis Crick without Franklin knowing. So this is a famous event in history. So, but what happens is Franklin got this very beautiful X-ray photograph, and Wilkins basically give this data to James Walton and Francis Crick. And this photograph basically means this. When you look at this pattern, it means DNA must be a helical structure. It's something like the alpha helix. It's a helical structure like this. This is one. Another thing is it has 2 periodicities. Periodicity means it has 2 features that appear again and again and again. One feature happens at 3.4 angstrom, and one feature happens at 34 angstrom. So something repeats every 34 angstrom, something else repeats 3.4 angstrom. This is what we can learn from this X-ray photo called photo 51. And another thing, well, then after that, James Walton and Francis Crick start to build a model like what DNA actually look like. And another thing they know at the time when they build the model is called the Chargaff rules. This rule basically says it has 4 different things. Well, the base composition of DNA generally is different from one species to another. That's probably not very surprising. Another thing is DNA from different tissues of the same animal, same species, have the same base composition. Base composition means every DNA has 4 base, A, T, C, G. So base composition means the percentage of A, percentage of T, percentage of C, percentage of G. And DNA isolated from different tissues of the same species have exactly the same ATCG components. Okay, that's the second thing. And the third rule is this composition does not change with an animal's age, nutritious status, or the environment. And final thing is actually very interesting. He found that all the purines, A plus G equals to C plus T. All the purines equals to the number of purines equals to number of pyrimidines. And the number of A equals to number of T. The number of C equals to the number of G. Okay. So this is what James Walton and Francis Crick knew at the time. The DNA must be a helical structure, and with something repeat every 3.4 and 34 angstrom. And in the DNA, somehow composition of A, the number of A equals to number of T, number of C equals to number of G. And of course, they know DNA is composed of nucleotide. This is all they know at the time. And after that, they built a model. Today we call that Walton-Crick model. This is the model look like. They think, well, DNA is a helix, but it's not one helix structure. It's two helix. It's two helix like this. And it's two strands. So the 3.4 happens, that's the distance between the base, okay? The base, the distance between the two bases is 3.4 angstrom. And so the helix is about every 10.5 base, or roughly 10 base per cycle. And that would make the total distance here is about 34 or 36 angstrom. Okay? And this is important. They think there are two strands. Okay? The two strands. In the two strands, if one strand has A here, the other strand must be T. And A and T can form two hydrogen bonds. And if one strand is G, the other strand must be C. And because G and C can form three hydrogen bonds. And this will explain the tag-F rules. Why in any animal's DNA, A always equals to T, C always equals to G. This is because it has two strands, and the base on the two strands are paired to each other. Okay. And this is what this model says exactly. So it has, it's a helical structure, and it has two DNA chains. It's a double strand. The double strand want around the same axis to form a right-handed double helix. It's right-handed, so it's going like this. And another thing is, well, the ribose in the phosphate group is on the outside, and the base is on the inside, because again, that's a hydrophobic feature. The base is hydrophobic, so it's hidden inside, and the phosphate ribose are hydrophilic, so they are outside. And this is very important. G always pair with C, form 3 hydrogen bonds. A always pair with T, and form 2 hydrogen bonds. And finally, the two chains runs parallel to each other. Parallel means this, anti-parallel. We know every nucleotide has a direction, 5' to 3'. Now you have a double strand. So the double strand, they propose that for the base pair to happen, the double strand must happen. The other strand goes to, 3' to 5'. The two double strands must be anti-parallel. This is very important. If one strand goes 5' to 3', the other strand must go 3' to 5'. This only when you go this way, you can form the right base pairing. Okay? So basically, their model says it's a double strand, and there's a base pair, and the double strand goes anti-parallel. Okay? And then the ribose goes outside, the base goes inside. And another thing is in their model, so because the two strand, when the two strand does not exactly symmetrical, the two strand is a little bit like this. So if you look at this graph, there will be a place where there's this place called major groove, and there are this place called minor groove. So there are some places the base is exposed more than other space. Okay, in the major groove, there's more base are exposed. In the minor groove, the base is basically hidden under the sugar backbone. And this is important because we will know that the base actually where the information is. If a protein needs to interact with DNA, normally it will interact at where the major groove is. Okay. And again, as we talked about in the last chapter, they think there are two modes of interactions between bases. One is the hydrophobic base stacking, one is the hydrogen bond. Okay. And then after that, they published 3 papers back to back to back in Nature. This is 1953. So Walton Crick published this DNA double helix. It looks like this. And Wilkins published the structure of an X-ray photograph. And Franklin published her photo 51 in a 3rd paper. And this event, this basically distinguishes between modern biology and the biology before that. So this is a milestone in biology because after that everything is different. Before this we only study like how things we only study metabolism, like how sugar is turned into energy. But after that, we know how proteins are made, and we can so say like if we can basically operate on the DNA level, which will change your protein. If we want a certain protein, we can change it. We can get the protein we want. If we want to get more proteins in something, in some cells, we can do that. If we want to delete something, delete some proteins, we can do that because now we know proteins, the information is in the DNA molecule, and the DNA we can do something about the DNA. We can change the DNA, we can delete the DNA, we can add in more DNA, some extra DNA we want. Okay, now another thing is, this is from their own Jason, Walton, and Crick's paper. Okay. We wish to suggest a structure for the salt of the oxyribonucleic acids, DNA. This structure has novel features which are of considerable biological interest. And here it says, it has been not escaped. Like this part is how the structure looks like. And after that, they say that it has been not escaped of notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what this means? This means, okay, you obviously you got something from your parents, right? So you look like your parents because you get something, and we again know that this something is DNA. Your parents basically give a copy of his or her DNA to you, right? And that also means there must be some mechanism, there must be some way to copy your parents' DNA to you. Also, in other words, there must be some way to duplicate, to replicate your parents' DNA and make a whole new DNA molecule. And that, because of the double-strand nature of the DNA, and the base pairing, this becomes immediately possible. Whenever you have two strands, one goes this way, one goes this way. 1', 2, 3'. Like say one strand on the DNA is A, T, C, G, A, T, okay? And by base pairing, the other strand must be T, A, G, C, T, A, okay? So say in your parents, it has two strands like this. And when you want to make a new DNA, basically you can do like this. You can take the two strands of DNA apart because they are bonded by hydrogen bond. It's not very hard to take them apart. After that, you can make a new strand here. The new strand has to go opposite way. So this is 3', this is 5'. The new strand, by base pairing, the new strand can make T, A, G, C, T, A. And you can have a new strand here, which should also go anti-parallel. So this is 5', this is 3'. You have A, T, G, C, T, A. And as you can see that now basically you can have from the old DNA, now you generated two new DNAs. Okay, now you have two new DNAs. The DNA can use a base pairing, it can copy itself and form a new DNA molecule. And this is not possible with proteins. Because in proteins there's no base pairing. Like one amino acid cannot make a copy of its own. Proteins cannot do that. Only DNA can do that. That's why DNA can be an information carrier. Okay, we'll continue next class.